Okay, so you have to use the sentence from, I don't know, the intro discussion part of it. Interestingly, it was recently demonstrated that BRCA1 positively regulates the expression of MAD2, another potent inhibitor of the APC activity, and those of other genes implicated in the mitotic spindle checkpoint, emphasizing BRCA1 requirement in the spindle assembly checkpoint control. So first question is, what does this sentence mean? I think the sentence means that BRCA1 has like it plays like another role other than DNA repair, which is to, I think it does have it does have something to do with the mitotic spindle checkpoint. And so I think its other role, I think the sentence means that it has another role, and that other role is to make sure cell division doesn't occur until everything's lined up at that mitotic spindle and it's all happy and good to go. Yeah, it's also saying that BRCA1 plays a part in MAD2 by regulating the expression of it positively. So what experiment would you do to show that BRCA1 also positively regulates the expression of MAD2? First you'd have to overexpress a BRCA1. And then you got to see the promoter then you have to check the promoter activity of MAD2 to see if positively regulating or overexpressing affected the expression of MAD2. And so, I think I'd do 3 plates. And then in number one, I think I just do cells with no BRCA1. Without BRCA1. And then, so for this one, you'd have to do an overexpression vector and then a luciferase assay. Luciferase vector, whatever it is. So for plate number 2, I would do, I think I'd just do cells with the luciferase. With lucifer. And then for plate number 3, I would do cells with the overexpression vector. And luciferase. So those would be the 3 plates. Again, doing an overexpression to raise the levels of BRCA1 in these 2 plates. These 2 would have BRCA1, this would not have BRCA1. And then these 2 would have this one would have luciferase, this one would have the overexpression and luciferase. Yeah. And then from that you would get a nice graph because of the luciferase assay. And so 1, 2, 3. I think that plate number 1 would have a very low, a low bar, or just no bar at all. Because BRCA1 regulates the expression of MAD2. And then I feel like plate number 2 would have a slightly higher bar, but plate 3, the one with the overexpression vector and luciferase, that would have a much higher bar because there would be, I'd say more MAD2. There's just an uppercase A, okay. So more MAD2 because of the overexpressed BRCA1 in that plate. because BRCA1 arrow up. Cool. So I'm pretty sure that's what it would look like. I don't think plate 1 would have any, or it would have very little MAD2. But plate 2 would have a little bit of MAD2, but plate 3 would have the most because of the overexpressed BRCA1. There's just a normal amount of BRCA1 in this second plate, so it would just be a normal amount of MAD2. Okay. Question 3, using the above results, draw results for flow cytometry in cells treated with Taxol for each plate. Okay, so we need 3 graphs. So 1, 2, 1, 2, 1, 2. Okay. Okay, so I'm pretty sure so this sentence is talking about the mitotic spindle checkpoint, which is obviously happening at M phase. And so I'm pretty sure the graphs would kind of look like this. It would be just the one hump. I think they would all look the same, or be very similar, but they'd all have that hump at the G2 M into M. So I'm pretty sure that's what, yeah, I'm pretty sure that the 3 graphs would look the same for the 3 different plates if they were treated with Taxol. Because there'd be, because of the more MAD2, yeah, because MAD2 is a potent inhibitor of the APC activity. The APC pushes it from metaphase into anaphase. And so if it's inhibiting the APC activity, that means it can't go from metaphase to anaphase. So it would stay in the G2/M. So yeah, it would look like this. Okay, I believe so. Yeah.